My group's research aims to identify potential targets for personalized multiple myeloma treatment. Multiple myeloma is a cancer which annually affects 30,000 adults. Firstly, let's talk about the interactions between cancer cells and the immune system. Cancer cells have certain small and large variants which differ from the standard human genome. These variants express specific proteins called neoantigens, which are later attacked by the immune system. By identifying a cancer cell's specific variants, we can predict the expressed neoantigen and provide personalized immunotherapy treatment. Most research done so far has been on small variants of a few base pairs. However, research regarding large variants, known as structural variants, is a decade behind. Structural variants play a large role in multiple myeloma, as 60% of multiple myeloma cases are attributed to structural variants between chromosome 14 and another chromosome. It is important that we develop tools to detect structural variants specific to each patient's tumor. Now let's discuss how we identified these potential targets. Firstly, DNA was extracted from 77 multiple myeloma cell lines and sequenced to generate hundreds of millions of tiny sequences that represent each cell line's genome. Because we can't handle this amount of data on our laptops, we analyzed these terabytes of data using the Texas Advanced Computing Center. To identify structural variants from this sequencing data, we used 4 recently developed tools. After running these tools, we compared these tools using various metrics such as runtime. Runtime across the tools varied quite dramatically. For example, this tool took less than a minute, whilst this tool took a few hours. Another property we analyzed was accuracy. To determine this, we were informed of known structural variants for each of the 77 multiple myeloma cell lines. This graph illustrates how the tools struggle to different extents to identify these known structural variants. Due to the tool's varying output, it made sense to focus more on the 32 structural variants detected by more than one tool. These plots show these potential rearrangements across multiple chromosomes. For example, this line here shows a potential structural variant between chromosome 1 and chromosome 2. These potential structural variants we have identified brings us closer towards the goal of targeting cancer cells with specific immunotherapy treatments. Thank you for listening.